Moving into phase 2, this is where we see our screening of new therapies. So phase 2, they've gotten through phase 1. I'm not killing that many people, that's good. Phase 2, I'm trying to still learn a lot about safety, but learn a lot more about my doses. I'm not going to probably look at final efficacy, but I might have some type of biomarker or surrogate outcome that I'm going to determine kind of a pseudo-efficacy that I'm going to power on. Our general idea in phase 2 is it safe enough and is there sufficient activity enough to bother to move this to phase 3, move these to the large pivotal trials? Again, if you look at the old definitions, they'll say, well, should we bother to move it to a randomized trial? These days, almost every phase 2 trial I work on is randomized. It is a rare exception that they're not randomized. Your issues of safety are still going to be really important here. And you have a fairly small number of patients. Might be several hundred, but it still depends. Like, I don't see that many that are more than 100 people per arm, but sometimes it happens. Your problems in phase 2 is that you are more likely to run into the unblinded or single-blinded studies here, have all the problems that we talked about at the beginning of the course. You have issues with placebo effect, investigator bias, regression to the mean. If you're running a single-arm study, you're not really sure what caused those changes to happen. That's the reason that you see this big push. So if you work in industry, if you're one of these pharmaceutical or biotech companies, you're running non-randomized trials, you're running unblinded trials, you tend to move into phase 3 and you get burned. Everything that looked fabulous in phase 2 does not pan out in phase 3. So now I've just wasted sometimes billions of dollars running those phase 3 trials. That's the reason that people are getting a lot tighter in phase 1 and phase 2, because better to stop the product early if it's not producing results. Same thing in academic medicine, folks. You're better to fail small, but be wise about your failures. And with that, then I'm going to talk to you about a non-randomized design, but actually randomized designs have been built out of this. So for phase 2, there's something called a 2-stage optimal design. This was developed out of NCI back, I think, in 1980, if I remember right. And the idea is the premise for a lot of current phase 2 designs. You want to seek to rule out an undesirably low response probability. So let's say only 20% of people respond to treatment. I'm going to rule that out in favor of something that shows useful activity. And let's say I've defined useful activity to be that 40% of my patients are stable or have improved outcomes. So in oncology, they might run a single-arm, 2-stage design using an optimal design and a predefined response definition. It is hard to define a response because then people want to tweak those numbers, right? But again, if we want to rule out a probability of 20% in favor of 40%, let's say I set my type 1 error at 0.1 and my type 2 error at 0.1, right? So I'm willing to make, this is one of those cases where I set them equal to each other. The idea being I'm willing to tolerate a type 1 error. If there's something there, though, I want high power. I really want to see it. So roughly, in this kind of more probability-based setting, I've got a 10% probability of accepting a poor agent and a 10% probability of rejecting a good agent. So if you look in Simon's paper, and if it's not posted, I will ask Daniel to post it up in your class segment. He has this paper with all these different kind of trade-offs of these different probabilities and varying alpha and beta values. This is something that Steinberg and Vinson then built upon in the early 2000s for randomized trials. So we're going to blow this up in a second. This table is table 1 from Simon's paper. we have the difference in the probabilities being 20%. And we've got that we're going to reject a drug if the response rate falls through here. There's something called the optimal design, which is what Simon was trying to promote, and one that it was already out in the literature called the minimax design. So let's look up here. Our null is a response rate of 20%. We want to actually see 40% responders. So what do I do here? What I want to do is initially I am going to enroll 17 patients. So if you looked at the tiny footnotes, we're going to be in kind of this top row here, the bottom row, the bottom chunk. So stage 1, I enroll 17 patients. If 0 to 3 of them have a clinical response, I stop accrual. I assume it's not an effective agent. So what I'm saying is there's no way I'm going to be able to rule out this 20%. So I'm just stopping early. But if 4 to 17 patients have a positive response, I can move on to the second stage. I'm going to accrue up to 37, so I'm going to accrue 20 more patients. And if 10 of the 37 have a positive response, if 11 or more of the 37 have a positive response, then I think it's an active agent. So I'm rejecting if I have 3 or fewer than if I have 10 or fewer. What these other numbers were are, here I have a chance I'm stopping early, right? It might be I stop at 17. If 17 out of 17, I'm done, realistically. But let's look at this. So if I do the standard sample size, under the null hypothesis, I have to enroll 26 patients. Probability I'm going to end under the null is 55%. It's pretty good. So here it is in words for you what I just said. Under the design, if the null hypothesis is true, there's a 55% probability of early termination. This is one reason we like phased designs, right? We get out early. It's not 100% sure, but 55% is better than 0%. On average, in that 2-stage optimal design, under the null, I enroll 26 patients. If I used a 1-sample test of proportions, I need 34 patients. So 34 is fewer than 37, but I don't have that chance to stop early. And that's what I'm gaining in this 2-stage design. If I use a 2-sample randomized test of proportions, I have 86 patients per group. And that's the reason you have this lure of a single-arm study. Why do I want to have 2 arms and randomized when I can get some information? Maybe it's a problem, but I have so many fewer patients. It takes me less time and less money. This is what you have to balance as an investigator. Again, there are a lot of newer methods available. They all end up citing Simon, so that's why Simon is here. So let's talk about historical controls now. In historical controls, let's say I've got a rare disease. I want to double their survival. Survival right now is 15.7 months, and I want to, with my new intervention, double that median survival to 31 months. I say I'm going to have a type 1 error of 0.05. I want a one-tailed test, and I want my power to be 80%. So why one-tailed? Basically, if they're dying, then this is going nowhere, right? So if my survival is less, no, that's it. So and also the reason that they went ahead and did a one-tailed test is they said, this is a rare disease, and we have to do everything we can to try to have a reasonable test. And because they're like, it's going nowhere, and they swore they were going to publish it either way, they're like, yeah, it's interesting, and it will be clear that this is wrong. So they had a one-tailed test. You need 60 patients for this, and we'll talk about how you figure this out. about 30 in each of the 2 study arms. They come back and they say, okay, Dr. Johnson, I can accrue 1 per month. It was actually a different doctor they were talking to, but he let me share the example. So if you can get 60 patients, 1 per month, that's 5 years. To just enroll them in your study, let alone follow them in the study. They needed 36 months of follow-up, so that's an additional 3 years. So now it's going to take me, in best case scenario, 8 years from the time I start enrolling patients to when I stop collecting data. Yeah, that's a long time to wait for an answer. Plus, realize it probably took you a couple of years to get this study up and running. It's going to take you a little bit of time to analyze and publish your data. So they said, well, we want to use historical controls. We have a bunch of patients. They were controls in other studies. We want to use their data and put all patients that we have on the new therapy and compare them to the old patients. So there was this old data set. We had 35 patients. They'd been treated in National Cancer Institute with this disease. Problem is they were initially treated from 1980 to 1999. Of those 35 patients, at the time they were looking at the records, 3 of them were still alive. Median survival time for the historical patients, so where they'd gotten the value from, was 15.7 months. And this is basically kind of like this observational study, right? But, 1980, it's 2015 now. There's a lot different. When we diagnose them, how we diagnose them, what measurements you're taking, how you're recording it, how you decide that people are progressing, the treatments that we're giving them, not just to treat their disease, but everything else, to help their nausea, to help their anything. It's all different. So you have to decide, do you use this data? Is it really controlling for everything you need to control for? Does anybody else have data? Can you work with people around the world to do this protocol? How do you do it? So these are the problems that come up in phase 2 studies. You've got to constantly be weighing the advantages and disadvantages of your study designs. You may have a single-arm study that has small sample size. That's good in some ways, but you've got no control information. You may have a 2-stage design. Again, you might stop early with a small sample size, but if it's a single-arm 2-stage design, you've got no control. You might have the correct responder, non-responder rules, or you may not. Are you sure that that difference is really what you care about? Can you make a binary distinction to really even define a responder? Historical controls. Again, it's great because you can use a small sample size, but you don't know how accurate that control group really is. You might decide to run multiple arms, sometimes randomized, sometimes not. Sometimes I see people run like 8+ arm studies, but then they have no control arm. They're just comparing all the different doses to each other. I'm like, that's great, but what if none of your doses work or all your doses work the same? Like, you don't have a way to compare. So if you're going to be running multiple arms, please, please, please have something with some control in it, because it could be that every dose does something, but it all does the same thing. The downside is it takes a lot more people. That said, I see more and more phase 1 and phase 2 studies enrolling hundreds of people because they want to make sure what they're moving forward into these final large pivotal trials is what they've got the best shot at, and that they better understand a lot of the safety information, the pharmacokinetics and pharmacodynamics, and a lot of other information about potential efficacy in trials, especially in multiple different populations, because that's where we do our trials.